Hello and welcome dear students. Today we are going to discuss about the topic models explaining maintenance of genetic polymorphism. The main objectives of this module are to know about the genetic polymorphism, to know about the types of DNA polymorphism, to know about the maintenance of genetic polymorphism, to know about the sickle cell anemia and malaria. Dear students, let's first discuss the genetic polymorphism. Genetic polymorphism is a difference in DNA sequence among individuals, groups, or populations. Sources include single nucleotide polymorphism, that is SNPs, sequence repeats, insertions, deletions, and recombinations. For example, a genetic polymorphism might give rise to blue eyes versus brown eyes or straight hair versus curly hair. Genetic polymorphisms may be the result of chance processes or may have been induced by external agents such as viruses or radiation. If a difference in DNA sequence among individuals has been shown to be associated with disease, it will usually be called a genetic mutation. Changes in DNA sequences that have been confirmed to be caused by external agents are also generally called mutations rather than polymorphisms. Polymorphisms arise through mutations. The mutation may be due to a change from one type of nucleotide to another, an insertion or deletion, or a rearrangement of nucleotides in the DNA. Once formed, a polymorphism can be inherited like any other DNA sequence, allowing its inheritance to be tracked from parent to child or offspring. Polymorphisms are also found outside of genes in the vast quantity of DNA that does not code for protein. Indeed, regions of DNA that do not code for proteins tend to have more polymorphisms. This is because a change in the DNA sequences that encode proteins may have a harmful effect on the individuals who carries it. Synonymous polymorphisms are those that do not have any effect on the organism and are said to be selectively neutral, as the substitution causes no amino acid change in the protein produced. This is also called a silent mutation. A non-synonymous substitution results in an alteration of the encoded amino acid. A missense mutation changes the protein by causing a change in the codon. A nonsense mutation results in a misplaced termination codon. One half of all coding sequences, SNPs, result in non-synonymous codon changes. DNA polymorphisms comprise three types, that is tandem repeat polymorphism, copy number variation, and single nucleotide polymorphism. For polymorphism to exist, new alleles must be both generated and maintained in the population. Many traits under selection have high heritabilities. One of the most striking examples is the surprisingly large genetic variance that exists for sexually selected traits. To explain the maintenance of such genetic polymorphisms, heterozygote advantage, frequency-dependent selection, and environmental fluctuations have been proposed. Balancing selection, in contrast to directional selection, maintains genetic polymorphism in populations. When some proportion of the allele with the lower geometric mean fitness is shielded from selection, frequency-dependent selection remains the most commonly invoked heuristic explanation for the maintenance of genetic variation. Dear students, let's now discuss the types of DNA polymorphism. The first one is tandem repeat polymorphism. DNA sequences that are repeated in tandem are common throughout the genomes of a wide range of species, including humans, and are often highly conserved, suggesting that they have an important function. Most research on tandem repeats has focused on those in the human genome, such as microsatellites, used as genetic markers and as repeat expansion mutations causing dominant or recessive disorders with Mendelian inheritance patterns. The term dynamic mutations has been used to describe the variation in tandem repeat length that has been found in many human genes to cause a range of repeated expansion disorders, particularly those affecting the nervous system, including Huntington's and other polyglutamate diseases, Friedreich's ataxia, and Fragile X syndrome. A much larger array of tandem repeats are present in and between genes that are not known to be involved directly in diseases of Mendelian inheritance. Tandem repeats are also referred to as simple sequence repeats, microsatellites, or mini satellites. These repetitive sequences can be located in exons, introns, or intergenic regions, providing opportunities for the modulation of gene expression and for that of the structure and function of RNAs and proteins. The term tandem repeats is used to describe tandemly repeated DNA sequences, also known as simple sequence repeats or satellite DNA, which includes both microsatellites and mini satellites. Tandem repeats can involve mononucleotides, dinucleotides, or trinucleotides, that is triplets, tetranucleotides, etc. Microsatellites range from 1 to 5 and 1 to 10 base pairs in length. Thus, tandem repeats with a motif longer than the upper limit for microsatellites, that is greater than 10 base pairs in length, are generally called mini satellites. Tandem repeat polymorphism or SSR polymorphism have also been referred to as variable number tandem repeats or repeat length polymorphisms. Dear students, let's now discuss the short tandem repeats. Short tandem repeats or STRs are very short stretches of DNA that are repeated back to back at various locations throughout the human genome. Typically, the repeating sequence is just 2, 3, or 4 base pairs in length, and the number of copies found back to back is variable across a wide range. Unlike the case for the DNA sequences of coding genes, there is no correct number of repeats for a specific STR in the genome. They are simply areas within the genome in which variation is normal and healthy. For any specific STR, each person will have 2 copies, one that is inherited from his/her mother at conception and the other that is inherited from his or her father. STRs or short tandem repeats are helpful in forensic and paternity testing. Because there are a lot of natural variations in STRs, the chance of 2 people matching for the exact number of repeats on both inherited copies of the STR is fairly small. On combined analysis of many STRs across the genome, the probability of 2 people matching by a random chance was found to be extremely low. Genetic polymorphism is defined as the inheritance of a trait controlled by a single genetic locus with 2 alleles in which the least common allele has a frequency of about 1% or greater. Genetic polymorphism is a difference in DNA sequence among individuals, groups, or populations. Sources include single nucleotide polymorphisms, sequence repeats, insertions, deletions, and recombination. Genetic polymorphisms may be the result of chance processes or may have been induced by external agents such as viruses or radiation. If a difference in DNA sequence among individuals has been shown to be associated with disease, it will usually be called a genetic mutation. Changes in DNA sequence that have been confirmed to be caused by external agents are also generally called mutations rather than polymorphisms. SNPs are the most common type of genetic variation in humans. Understanding the functions of SNPs can greatly help us understand the genetics of human phenotype variation, especially the genetic basis of complex human diseases. Detection of these polymorphisms can be carried out by different methods, including allele-specific PCR, restriction fragment length polymorphism, the microarray technique, and whole genome sequencing. Copy number polymorphism or variation. Genetic association studies generally evaluate SNPs, which are variations in single nucleotide at specific genomic locations between individuals of the same species. Recent results indicate that human genome contains another frequent type of polymorphism, that is copy number variation or CNV. A CNV is a variation in which a segment of DNA can be found in various copy numbers in the genomes of different individuals. CNVs range in size from a few hundred nucleotides to several megabases. Compared with SNPs, CNVs affect a more significant fraction of the genome and arise more frequently. Hence, CNVs significantly contribute to human evolution, genetic diversity, and an increasing number of phenotypic traits. CNVs of DNA sequences are abundant in natural population and are functionally significant but still needed to be fully ascertained. A CNV is generated by both recombination and replication mechanisms and a de novo locus specific mutation rate, which is higher than in SNP. Let us now discuss the single nucleotide polymorphism. A single nucleotide polymorphism is present at a particular nucleotide site. The DNA molecules in the population often differ in the identity of the nucleotide pair that occupies the site. For example, some DNA molecules in the same population may have a TA base pair at a particular nucleotide site, whereas other DNA molecule in the same population may have a CG base pair at the same site. This difference constitutes an SNP. The SNP defines two alleles for which there could be three genotypes among individuals in the population: homozygous chromosomes or heterozygous chromosomes with TA in one chromosome and CG in the homologous chromosome. SNPs may arise from a substitution of one nucleotide by another at the polymorphic site. Substitution can be transitions. A transition is the replacement of one purine nucleotide by another purine nucleotide or one pyrimidine by another pyrimidine base. A SNP may also be a single base insertion or deletion variant referred to as indel. A synonymous codon change or silent mutation single nucleotide polymorphism is one that does not result in a change of amino acid because of the degeneracy of the genetic code. A substitution that changes a codon coding for one amino acid to a codon coding for a different amino acid is referred to as a missense mutation. A nonsense mutation is a type of non-synonymous codon change in which a stop codon is formed, thereby leading to premature termination of a polypeptide chain and formation of a truncated protein. SNPs are responsible for a disease do not necessarily have to occur in coding regions. That can occur in, for example, any genetic region that can ultimately affect the expression, structure, and activity of protein encoded by a nucleic acid. Such genetic regions include, for example, those involved in transcription. Examples are SNPs in transcription factor binding domains, in promoter regions, in areas involved in transcript processing, such as SNPs at intron-exon boundaries, which may cause defective splicing, or in mRNA processing signal sequences, such as polyadenylation signal regions. Dear students, let's now discuss the maintenance of genetic polymorphism. Many factors act to increase or maintain the amount of genetic variation in a population. One of these is mutation, which is in fact the ultimate source of all variation. The theoretical basis of mutation variation is simple. Mutations will maintain variation if mutation rates are sufficiently high. Empirically, deleterious mutation rates can sometimes exceed 1 per individual per generation, and it is well established that transient mutations contribute substantially to standing genetic variation in fitness-associated traits. However, mutations do not occur very frequently, only at a rate of approximately 1 mutation per 1 lakh or 10 lakh genetic loci per generation. This rate is too slow to account for most of the polymorphisms seen in natural populations. However, mutation probably does explain some of the very rare phenotypes seen occasionally, such as albinism in humans and other mammals. A polymorphism can only be maintained by fluctuating selection when some proportion of the allele with the lower geometric mean fitness is shielded from selection. Such cases are given when the recessive homozygote has lower geometric mean fitness but higher arithmetic mean fitness than the heterozygote. When the heterozygote has higher geometric mean fitness than both homozygotes, when selection fluctuates both in space and time, or when some genotypes are shielded from selection in a seed bank. As sex-limited traits are also partly shielded from selection, they are expressed in one sex only. Have already mentioned that fluctuating selection should be able to maintain a polymorphism for traits with sex-limited expression. Many sex-limited traits involve interactions between individuals, and such traits should be especially prone to be influenced by fluctuations in population density. Temporal change in population density and almost ubiquitous observation will probably cause strong fluctuating selection on sex-limited traits. A male with a superior but costly courtship signal might be example at a disadvantage under low population density because only few females are available and the cost of the signal outweighs the advantage of his attractiveness, whereas under higher population density this male might achieve many copulations and the benefit of attractiveness may thus surpass the cost of signal production. Similar arguments can be made for intrasexual competition, mechanisms of female choice, or for behavior used in offspring care. Fluctuating selection and sex limitation of traits are thus both widespread and likely to occur, and the proposed effect of fluctuating selection on the maintenance of genetic variance should be relevant for a large number of traits or characters. A second factor contributing to genetic variation in natural populations is selective neutrality. Selective neutrality describes situations in which alternate alleles for a gene differ little in fitness. Because small fitness differences result in only weak natural selection, selection may be overpowered by the random force of genetic drift. Alleles whose frequencies are governed by genetic drift rather than by natural selection are said to be selectively neutral. Under neutrality, allele frequencies vary over time, increasing or decreasing randomly. Over long periods of time, random fluctuations in the relative frequencies of different alleles may result in some being eliminated from the population. However, genetic polymorphisms are long-lived and novel neutral alleles may arise continually through mutation. Finally, several forms of natural selection act to maintain genetic variation rather than to eliminate it. These include balancing selection, frequency-dependent selection, and changing patterns of natural selection over time and space. The contribution of balancing selection to standing genetic variation can also be substantial. In a particularly striking example, sequencing studies in temperate Drosophila populations have found hundreds of polymorphic loci which undergo seasonal oscillations in allele frequency. Many of these polymorphisms appear to be ancient, suggesting pervasive long-term balancing selection. Balancing selection occurs when there is heterozygote advantage at a locus, a situation in which the heterozygous genotype, one including two different alleles, has greater fitness than either of the two homozygous genotypes, one including two of the same allele. Under heterozygote advantage, both alleles involved will be maintained in a population. Fundamentally, stable polymorphism requires negatively frequency-dependent selection, that is, low-frequency alleles must be favored over high-frequency alleles. In temporally and spatially uniform environments, instances of negative frequency dependence are largely restricted to immune system genes. Batesian mimicry and self-incompatibility loci. Heterogeneous selection substantially broadens the types of loci potentially subject to negative frequency dependence, since the latter only needs to arise when averaged over the environmental heterogeneity rather than within each generation or within spatially homogeneous subpopulations. A classical example of heterozygote advantage concerns the allele for sickle cell anemia. Dear students, let's discuss the relationship between sickle cell anemia and malaria. Sickle cell disease, also known as sickle cell anemia, is a potentially deadly genetic disease that causes the red blood cells to become misshapen, preventing enough oxygen from being delivered to all parts of the body. Having two of the same allele of a given gene means an individual is homozygous for that particular gene. If the alleles are not identical, then the individual is heterozygous for that particular gene. In areas where the malaria parasite is present, individuals who are homozygous for the sickle cell allele who will get sickle cell disease, and individuals who are homozygous for the normal hemoglobin allele who can contract malaria, both have a selective disadvantage. In areas where the malaria parasite is present, individuals who are heterozygous for the sickle cell allele are at a selective advantage because they are protected against malaria but do not get sickle cell disease. In areas where the malaria parasite is present, individuals who are homozygous for the sickle cell allele, that is, who will get sickle cell disease, and individuals who are homozygous for the normal hemoglobin allele, that means who can contract malaria, both have a selective disadvantage. A single base substitution actually causes the sickle cell allele disease. The mutation that causes sickle cells also compromise the ability of the malarial parasite to reproduce. Individuals who are homozygous for the sickle cell allele have sickle cell anemia, which causes the red blood cells to become sickle-shaped when they release oxygen. These sickle-shaped cells become caught in narrow blood vessels, blocking blood flow. Heterozygotes, however, have normal donut-shaped blood cells and do not suffer from sickle cell anemia. In addition, they enjoy a benefit of the sickle cell allele, which offers protection from the disease malaria. Consequently, heterozygous individuals have greater fitness than individuals who have two copies of the normal allele. Heterozygote advantage in this system is believed to have played a critical role in allowing a disease as harmful as sickle cell anemia to persist in human populations. So evidence for this comes from an examination of the distribution of the sickle cell allele, which is only found in places where malaria is a danger. Another form of natural selection that maintains genetic variation in populations is frequency-dependent selection. Under frequency-dependent selection, the fitness of a genotype depends on its relative frequency within the population, with less common genotypes being more fit than genotypes that occur at high frequency. Frequency-dependent selection is believed to be fairly common in natural populations. For example, in situations where there is competition for resources, individuals with rare preferences may enjoy greater fitness than those who have more common preferences. Frequency-dependent selection may also play a role in predation. If predators form a search image for more common prey types, focusing on capturing those, less common phenotypes may enjoy better survival. Finally, changing patterns of selection over time or space can help to maintain genetic variation in a population. If selection patterns fluctuate over time, different alleles or genotypes may enjoy greater fitness at different times. The overall effect may be that both alleles persist in a population. So changing selection pressures over time are encountered by a species of grasshopper characterized by two color morphs, a brown morph and a green morph. Earlier in the year, when the habitat is more brown, the better camouflaged brown grasshoppers enjoy greater protection from predators. Later in the season, however, the environment is greener and the green grasshoppers have higher fitness. Another possibility is that selection patterns vary from one place to another as a result of differences in habitat and environment. The prevalence of different genotypes in different habitats combined with gene flow between habitats can result in the maintenance of multiple alleles in a population. Dear students, it was all about today's lecture regarding models explaining maintenance of genetic polymorphism. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.